Yeah, unfortunately, Klaus here that couldn't make it for this meeting, so I will give the talk instead. We have recently witnessed a plethora of connectomes that have become available and concern not only the human brain, for instance, through the Human Connectome Project, but also the brain of other animals, both vertebrate and invertebrates, like mice and Drosophila. And what is obvious in these connectome reconstructions that have been obtained in the last decade or so is that we have to do with a really intricate wiring pattern that characterizes the brain. And despite definitely some progress that has taken place in the field of network neuroscience, we still lack a comprehensive set of principles that distill the essential features that pertain to these brain connectomes. And we should do this distillation of principles in such a way that they are neurobiologically interpretable, and also in such a way that can potentially uncover a unifying thread that binds together all these seemingly diverse connectomes across all the species, so to uncover unifying wiring principles. And also, which is very important but rather neglected, is the ability to have some candidate mechanisms of connectivity formation that can give rise to this intricate wiring pattern that we observe in the brain. So these are the challenges that can be summarized that despite a wealth of data, we have no unifying principles or mechanism of wiring formation. And that is what motivates the approach that I will highlight in this talk. This is the diagram that highlights the conceptual arc of the approach. We have brain connectomes, let's say the raw data, that we aim to understand. And then we proceed to a distillation of the wiring principles that parsimoniously describe the essential features that pertain to these brain connectomes. And we will distill these principles in a neurobiologically interpretable way because we want these principles to function as a handle in order to have an idea of what potential mechanism of wiring formation can give rise to these intricate patterns that obey these principles. We will occupy ourselves with two basic features of brain connectomes, that is connectivity strength and heterogeneity and higher order network topology. What is the case in recent studies and also in a bit older studies is that brain connectomes, the connections in the brain are largely heterogeneous. So certain connections are very strong, whereas others are very sparse, and they can differ up to 5 orders of magnitude. And this connectional heterogeneity now calls for an explanation that can parsimoniously capture this heterogeneity. And in that respect, among the many concepts that we have inherited from Ramon et al., what is pertinent here is the concept of the wiring cost. So as Rick briefly iterated as well, this principle postulates that neural systems, including brains, are wired in such a way that regions that are close by in space are more likely to be strongly connected, whereas regions that are further apart in space, if at all, will be sparsely connected. And we see this here in the mouse study where already like 2 decades ago, where we have the span of the majority of the connections in mid to short range distances. And also in more recent studies in the macaque cortex, we see that there is a decay of strength of connections with physical distance. And that was we see here also in the scatter plot, apart from showcasing the importance of wiring cost, this residual variance that we observe dictates that there are additional principles that should be disclosed in order to have a more thorough understanding of the heterogeneity of connections. So, as it is usually the case in network neuroscience, we get inspiration from other fields of science, in that case, social networks. And what is useful for our purposes is what has been highlighted for social networks, namely the homophily principle. The homophily principle demonstrates the following. Given a social network, if I have close friendships with 2 people, then these 2 people will also become close friends themselves, as we might have experienced in our lives. But if I have just some loose relations, some acquaintances, let's say, then these acquaintances, it's not so likely that they will also form a friendship. So that means the more common people that I know, the stronger our connection will become. And we incorporate this concept now to understand brain networks. And for instance, in the case of the human brain, we see here that the larger amount of areas that two areas connect to, so the common neighbors, the more common neighbors they have, the higher the strength of the connections in between these areas is, as we observe here, like there's a sharp increase. And as we would expect from the wiring cost principle, distance plays a role, as we see here, but it has the opposite effect. With increased distance, we have a decrease of strength. And this phenomena, these two principles, characterize the connectomes of the mouse, the monkey, and the Drosophila connectomes. We have exactly the same behavior. So these results dictate the following, that despite the fact that we have brains that differ vastly in terms of brain size, for instance, or like if we pay attention to the human and the Drosophila brain, they have been separated, the species, more than 700 million years ago. And for instance, Drosophila contains around 100,000 neurons, but the human brain has 86 billion neurons. But despite these large discrepancies in neuronal numbers that the brain contains, these results dictate that these neuronal populations wire with each other by obeying the same set of principles, that is the principle of homophily and wiring cost. These results are helpful in order to parsimoniously describe the strength heterogeneity across species, across connectomes of different species, but they also pose a conundrum because they call for an explanation at the mechanistic level. That is, how a brain knows, quote unquote, to get wired economically, so to get wired in a way that it obeys the wiring cost principle, and also how a neural system and the brain in general can get wired during autogeny in order to obey the homophily principle. So now we will tackle the issues at hand from a mechanistic standpoint. And in order to do so, we will adopt computational modeling, so in silico approach. And in turn, this in silico approach is based on the depicted neurodevelopmental phenomena. So what is the case? What we see here is in the mouse brain, but this holds for the brain of other mammals and also invertebrates. The brain during development has a mode, a spatiotemporal mode of development. That is, neuronal populations migrate and populate different parts of the brain at different time points. So we have to do with spatiotemporal gradients that emanate from the origins that are highlighted here in gray. So we simulate this phenomena in a synthetic 2D brain. It's an abstract form, but as you will see, it's sufficient to sketch out the mechanism of interest. So we have origins of neurogenesis that successively populate the brain, the synthetic brain, at different time points. And as simulations unfold, as you see here, at different time points, more and more neurons populate the brain in an heterochronous and spatially ordered manner. And when a neuron populates its part of the brain, then it forms a connection, but it does so with no information whatsoever. It's purely agnostic to anything that we have observed in the empirical connectomes. It forms the connection purely on a stochastic fashion. So it chooses a direction, it develops an axon. And if it hits an available partner, another neuron, they form a connection. So this spatiotemporal unfolding of neural migration also leads to the formation of a connectome. And in that scenario, space and time correlates during development. And in order to examine the synergy of these two fundamental dimensions, space and time, in shaping the intricate patterns of the brain, we have a scenario where we detach space and time correlation. So we have here, as you see, a network that develops in different time points. So it has this heterochronous mode of development, but the neurons are spatially random. And we have a case also where we have a tautochronous scenario. So that means all neurons are simultaneously present in the brain and they form connections. And in order to compare the synthetic connectomes with empirical connectomes, we have to construct a region-to-region matrix, and therefore we parcellate the brain in artificial regions, and we form a region-to-region connectivity matrix. What we're interested in replicating with this model is what we have iterated in the first part, namely the relation between connectivity strength, homophily, and distance. And this relation in the empirical connectomes, in that case the human brain, is depicted here by this coefficient of determination that describes the relation between homophily distance to strength, so how much of the variance is explained by these two principles. And as you see here, the connectomes that have been developed in an heterochronous but spatially ordered manner, with deep green here, the bar, mimic quite a large part of this relation that we observe empirically, and it captures best this relation compared to the other modes of development. And exactly the same relation holds for the other species, and on average we have an 83% replication of the relation of interest, strength, homophily, and distance. So let's now change gear and address briefly some network topology metrics. We all have witnessed recent studies in the last decade or so that show that brain connectomes of human but also non-human animals have a non-random topology. For instance, they are characterized by community structure or modules, as you might have heard of, or they have a core. You might have heard also like the term core periphery. So now we want to test if our parsimonious model that is based on neurodevelopmental gradients can capture these more high-order network topology patterns. So in order to do so, to find out to what extent we can do that and what we can learn about empirical connectomes, we profile with commonly used metrics like modularity and clustering coefficient and so on, the synthetic connectomes that have been generated under these three different scenarios that we have seen: heterochronous, spatially ordered, heterochronous, spatially random, and tautochronous. And then we have a multidimensional space that characterizes the topology of all these synthetic connectomes. And this multidimensional space is collapsed in a 2D space, as you see here, that can be conceived as a network topology morphospace. And as you see here, in this morphospace, the first component describes almost all the variance between these two groups, meaning that the major differences that are observed in this synthetic connectomes that have been developed under different scenarios is the separation between connectomes that have been developed in a tautochronous fashion and connectomes that have been developed in an heterochronous fashion. But what we are really interested, this is an intermediate step, is to ask the following question. Given that we have these modes of development that we have simulated, what is the case with empirical connectomes? What type of development do the empirical connectomes mostly resemble? And in order to answer this, we use this 2D space, this morphospace, as a classification space, and we profile all the empirical networks with the same metrics that we have used in order to profile the synthetic connectomes. And the result is the following. The classification assigns almost unanimously all the brain connectomes of human and non-human animals in the heterochronous and spatially ordered scenario. So that means, given that we have this adult intricate wiring pattern characterized by the topology that we just iterated, most likely they come from a process, from a generated model, an endogeny mode, if you will, that is heterochronous and spatially ordered. So the above demonstrate now, again, at the cross-species level, transcending the vertebrate and invertebrate boundary, that all these seemingly diverse connectomes can be understood as a result of a parsimonious neurodevelopmental mechanism that is based on the principle of heterochronicity, so the development across distinct time points. And conjointly, what has been presented demonstrate that indeed we can distill unifying wiring principles that pertain to human but also non-human animals, so we have to do with unifying principles from that respect. And also we can uncover a parsimonious mechanism of connectivity formation that, again, as the results show, it has a universal scope transcending the vertebrate and invertebrate boundary. Thank you very much.